Next part, describe the lab purpose of this. Okay. So the purpose of this is to find out
Hi there. I'm just Montoya. And I'm Michael Hahn.
And I am Connor Kraus.
And this is Loyola Apes. Hello. Now, for this experiment, we are going to be monitoring the factors that affect the production of oxygen throughout photosynthesis. So, to do this, we are currently using a computer, a GLX, a photosynthesis tank, and the sun in the form of a light.
So, we use the light to give the plants in the photosynthesis tank the opportunity to go through photosynthesis. And then the sensors in the tank come back to the GLX, and the GLX puts the data onto the computer where we can look at it and interpret the data.
I'd just like to give you a little bit of background on photosynthesis. As you probably know, photosynthesis is a process in which light from the sun, or in this case, the lamp, enters into a plant and is used to produce oxygen. For this reason, I believe the more plants that are inside of a photosynthesis tank, the more oxygen will be produced. That is the hypothesis for our experiment, and after we record our results, we will see whether that proves true.
Here are the results for our experiment. The first run here, shown in orange, shows the temperature. It started out at a little bit below 23 degrees Celsius and fluctuated a little bit and ended a little bit above 23 degrees. Here is our dissolved oxygen. At the beginning, it was at a little bit above 4, but after we put in the plants, it dipped and eventually became quite a bit lower than 4. So what we think happened is that because we put 3 plants in our photosynthesis tank, they got too crowded and eventually started taking up too much oxygen from each other. So that explains why our oxygen graph slowly decreased over time.
Oh, well, we believe that a better way to finish up the experiment would have been to use a bigger tank, you see. As my friend Michael described earlier, we think that with so many plants crammed into such a small area, that they could not produce as much oxygen because they were not receiving the same amount of sunlight. So, if we were used to use a bigger tank, then the plants could no doubt receive more sunlight because they would not be so hemmed in, and therefore produce better oxygen levels.
Finally, the experiment can be made just a little bit better if you add more types of plants, different types of sunlight, and perform the situation repeatedly. In this way, you can ensure that you'll get the most accurate results for future experiments.
Well, we believe that this experiment proves that sometimes less is more. You know, with all these plants in there, the plants couldn't produce the optimum levels of oxygen. That's indeed. Indeed. Thank you.
Look at that. Yeah, what a good experiment.
I am Adam Laskowski, and this is our photosynthesis lab experiment.
And I'm Aaron Johnson. The purpose of this experiment is to see what factors affect the rate of oxygen production in photosynthesis.
Acting as the sun, the lights represent the energy of the sun. A large rubber stopper holds the dissolved oxygen sensor. It detects the oxygen levels in the water of the inner tank. The stopper also holds a stir plate moving at moderate to high speed. This will help us measure the temperature of the water as well as the oxygen levels.
The process of photosynthesis is when a plant takes carbon dioxide and water in the air, converts it into oxygen gas, O2, and glucose, or sugar in layman's terms. Photosynthesis is a process in which a plant or producer takes carbon dioxide from the air, water from the ground, and sunlight to produce oxygen gas and glucose, which is stored in the plant.
Photosynthesis is the reaction of a producer to sunlight, carbon dioxide, and water.
It then produces, as a result, oxygen gas and glucose, as shown in this picture.
As we can see, originally the allodial technical level exhibited significantly explain the amount of oxygen. When we started the stir bar up again, another experiment which we could conduct.
Adam, what do you predict for this experiment?
I predict that when we add the Alodia plant, the oxygen concentrations will increase because the plant is producing more oxygen.
So Adam, what are the results?
The results are, when we added the Alodia plant, the oxygen concentration actually decreased while the temperature increased.
Wow. How would you explain that, Adam?
Well, we don't really know why the oxygen decreased, but I predict that the reason why is because there's a foam top at the top of the Alodia plant that might have absorbed the oxygen as the experiment was happening.
The blue line on the bottom shows.
So Adam, if we did another experiment, what could we do to get better results or better demonstrate the principle of photosynthesis?
Well, I think instead of having a short experiment like this, we could have an experiment that lasts several days that measures the growth or decrease in the oxygen concentration in a large body of water.
And what do you predict the difference would be?
I think that there would actually be an increase in oxygen rather than a decrease.
Intriguing. So, how would you conclude this photosynthesis lab?
Well, I think that this lab was not a failure. Instead, it taught us that the Alodia plant does not produce oxygen when it goes through photosynthesis.
And you think that's why the oxygen levels were so low?
Yes, I think it consumed oxygen during photosynthesis, not produced it. Go.
The purpose of the experiment is to see what factors affect the rate of oxygen production in photosynthesis. The lights represent the energy of the sun. In the actual experiment itself, a large rubber stopper holds a dissolved oxygen sensor, which detects the oxygen levels in the water of the air tank. It also can measure the temperature. The stopper also holds a stir plate moving at a moderate to high speed, which will help us determine the temperature. In doing this, we will understand the effects of photosynthesis when we put in a plant.
Hi, I'm Edwin.
I'm Will.
This is Will. And we're doing the photosynthesis lab for Loyola Apes.
For our experiment, we're using the Data Studio on the Mac.
We'll put it here and
As
say
you can
like
see,
we're predicting that.
the temperature is going slightly up, and the dissolved oxygen is below what it was before the plants were inside. Let's see.
That's not what we're supposed to talk about. Okay, so for today's photosynthesis lab, we are using Data Studio on this laptop. It's a program that records the dissolved oxygen level and temperature through the GLX. And the GLX is connected to the oxygen sensor.
Is this thing?
Yeah, and it's placed on top of the stir plate, which stirs.
This is the stir plate. As you can see, this thing right here, it's making it spin. And we're using a fluorescent light right behind it, so...
As a light source.
Yeah.
The purpose of this lab is to measure the difference in dissolved oxygen when there's a plant inside the water and without plants in the water. And to measure temperature change using plants. Okay, so photosynthesis is a process that all plants go through in order to make their own food. They convert carbon dioxide into various sugars.
So this is glucose.
So this is the equation. Well, you can't really see, but it says carbon dioxide plus water with light equals sugar and oxygen. So basically, photosynthesis converts carbon dioxide into organic compounds, especially sugars, using the energy from sunlight. So photosynthesis occurs in plants, algae, and many species of bacteria, not in archaea. photosynthetic organisms are called photoautotrophs, since they can create their own food.
And as you can see, the temperature
Photosynthesis is vital for all aerobic life on Earth, as well as maintaining the normal level of oxygen in the atmosphere. Nearly all life either depends on it directly as a source of energy, or indirectly as the ultimate source of energy in their food. Okay, so the prediction of this lab, we predicted that the dissolved oxygen level will slowly rise, since we think the plant will give off more oxygen. And the temperature, we think that it's going to go down, since the plant will absorb heat rather than to release heat.
Yeah.
So this is our results for the photosynthesis experiment. This is the dissolved oxygen level. The red is run number 1 without the algae, without the aquatic plant. The run number 2 is with the aquatic plant and the light source.
As you can see in the second graph, this is the temperature. The temperature for the first run was it was increasing slightly, and it was the green. And the second run, which is orange, it actually rose a few kelvins, a few Celsius.
Yeah, a few levels of Celsius. And orange is the run with the aquatic plant.